Hello guys, once again, we are gradually approaching to the calculatory aspect of this lesson of microbial growth enumeration and growth parameter. We are now in lesson 3 where we'll now look at the microbial growth curve and growth parameters. And we have already discussed what microbial growth is all about. It's an irreversible increase in microbial cell population, and this cell population can be measured directly or indirectly, as we have taken in the first and second lecture, especially in the lecture serial number 2. And if you look at this graph, there is a graph that we are looking before us in the screen, and this graph is have a dome shape, like a bell shape, with a four peculiar phases, four peculiar regions that we are going to look at: region A, region B, region C, and region D. There is another dotted line here which we will explain, and this region we classify them as phases of microbial growth, and there are basic things that happen within this level that really that is going to define our calculation. If you look at this graph, in the horizontal, in the vertical axis, you will see microbial load. That is the measuring parameters. You can measure with either the substrate concentration, you can measure with product concentration, or you can measure with the microbial biomass, which is represented in M. These are the three parameters you can use to measure microbial growth in any microbial system, while you measure it over time, and the time is usually in minutes or seconds. Then let's go to the main business of the day. In the phase 1, which we call the lag phase, in this lag phase, this is the phase where the microorganism does not initiate or go into any particular growth activity. This is a period where the microorganism try to familiarize with the substrate and environment to know if there is enough nutrient, if the nutrient is what it can be able to utilize and metabolize, if the environment is conducive, if the temperature is optimum, if the pH is optimum, and if water activity is rightly in its proportion, in order to enable the microbial growth. If there is water activity, in order to kickstart the enzymatic activity that will initiate the growth. So if this phase is not successful in any microbial growth phase, the microorganism will not initiate into growth. If either it dies or it goes into survival mode, which we will discuss in this dotted line. So as soon as a microorganism is able to overcome this phase and able to find a way to overcome it, it will initiate into an exponential growth phase, which is the phase B, starting from this point, ending of this point to this point. This we call exponential growth, or what we call the log phase. This is where the microorganism initiate or go into high level of activity that multiplies the cell population over time in exponential order. And this is the phase where the calculation will all be discussed about. All the calculation will be discussing at this primary level, at this elementary level or starting up level. This is the only place we are going to discuss the microbial growth curve, which is the relationship between the initial here and the final stage of this exponential phase is given as 2 raised to power n. That is exponential growth. We will go into that discussion in lesson 4. So have that in back of your mind. That means at this phase, okay, we'll discuss it in a different slide in details for this. For this phase, which seems very flat and horizontal in the phase, we call the stationary phase. This is where the microorganism, now the death rate and the growth rate are at equilibrium. We also call this phase the steady state phase. That means the growth rate, the population that is growing and the population that is dying are at equilibrium. They are relatively at equilibrium, and this actually happens when the nutrient is almost getting exhausted. As soon as the nutrient can no longer support the rate of growth here, or the environment or the condition is changing due to debris or waste product that is now producing a feedback or trying to inhibit the general microbial activity, this growth phase will now, the growth population of the microorganisms within the system, closed system, will start declining. That means the death rate, the number of microorganisms that is dying over time, is greater than the number of microorganisms that is splitting or increasing over time. The growth rate and death rate is not at equilibrium. Now the death rate is higher than the growth rate, so the growth curve, the growth population start declining. It will decline to a point whereby the substrate or the nutrient or the microbial activity can no longer be sustained by the available nutrient, by the available environmental condition, by the available other genetic factors or factors that initiate or control microbial growth. This microorganism will go into survival mode. This survival stage is what we call the dormant stage, where we now this microorganism forms what we call protective coating, which we have discussed in the lesson 1 about spores. Once the microorganism experience a very harsh and unfavorable condition which they want to sustain the generation, and they do not have the characteristics in terms of environment, in terms of nutrients, in order to support it, the microorganisms will go into this phase of spore forming in order to safeguard the generation until the condition becomes favorable. It will initiate into vegetative cell and explode into another exponential growth phase. So looking at this graph, we tend to define this term, these growth parameters. These are the three basic parameters. Growth parameter, measuring parameters, we use a microbial analysis. We have the substrate, that is the rate of substrate depletion, the rate at which the microorganism utilize the substrate. You can use that as a means of measuring, as a measuring parameter to measure the growth rate of microorganism. Another one parameter you can use is the product concentration, the rate at which the microorganism produce a particular product, a particular byproduct. Is now, you can now use it to measure the rate of microbial growth, just like in ethanol production, where microorganism can produce ethanol. If you can measure the rate of alcohol production or ethanol production within that system, that is the product, you can use it to measure the rate of growth in microbial growth. Then another one, you can use the microbial cell concentration. This one is what we just discussed in the second lesson, where you say dry mass. So you can use this, the microbial biomass, the rate of biomass and cell increase, that rate. You can use it to measure that. Then you also have the parameter of time. You have time is a dynamic factor that changes over time and can be measured in minutes or seconds. The next slide, which we now extend the log phase, you can now look at the log phase is the initial phase. There is no actually microbial growth. That is where the microorganism tends to adjust to the new environment and now initiate growth. So what basically happens there is that the cell adaptation occurs here. The microorganisms are able to adapt to the environment before it can go. There is also a minimal change, which we also look at insignificant, not in terms of population now, but in terms of size and other cytometric factors within the cell membrane, in order to initiate the binary or exponential growth. That's why you call it minimal. It's not actually a growth to say in terms of microbial cells, but in a bigger cell we can define it as growth. But a microbial cell, we just look at increase in population, because the difference between the parent and the daughter cell, which we now split in, there is no significant difference. So that's why we call it minimal growth. The next phase we are going to explain is the exponential phase. This is where the cell goes into an astronomical increase by the relationship of 2 raised to power n. So at this point, the growth phase is higher than the death phase. So this is where you have the maximum growth rate of microorganism. Cell division, the slope goes are very steepy, very up, as we have discussed in phase 2, phase B. Then you also have here, you have where the nutrient utilization really occur. This is where the microorganisms utilize the substrate that is available. Then the next slide, we now discuss about the stationary phase. This is the phase, the growth phase is level off. Death phase and growth phase are at equilibrium. This is also known as equilibrium stage. And also the metabolic adaptation here tend to be alternating front and back for positive growth and negative death. So at this point, they are at equilibrium. The last phase is the death or decline phase. This is where the substrate can no longer, or the system can no longer sustain the high population of microbial cells. Therefore, definitely the cells will start dying more than the new ones that are coming up. So at this point, once this stage can no longer be sustained, it will initiate into survival mechanism, which we have discussed before, formation of spores to survive the harsh condition. So with this, you will understand what we really mean by microbial growth curve. And this ends our lesson. 3. this ends our lesson 3 and we'll move into the next lesson 4. I would like to end the video here so that we'll have a in small bits so that we can be able to upload it and the data can be well managed. Thank you.